Okay, so what we have here is just a review of what we already went into. So you've had action potential, so your conscious control to move them and contract the muscle has come down the axon and down to the axon terminal. So now we'll look at your neuromuscular junction, and when you do, you'll see that there's these vesicles with acetylcholine inside. When action potential reaches this region, the vesicles will come down and release through exocytosis. Acetylcholine will cross the synaptic cleft and attach to these receptors. When they do that, they change the charge of your sarcolemma, causing sodium, which is always surrounding your neuron, to cross the synaptic cleft and start entering inside of your muscle fiber, changing the charge again, and then the potassium that's inside of your muscle fiber will move within and therefore out of your muscle fiber. Okay. Now the action potential is inside of our muscle, we're going to go on next. And essentially what's going to happen is so your action potential is going to release some other steps in order to contract the sarcomere below. So just a reminder about your sarcomeres. Your sarcomeres are made up of myofilaments, actin, which is your thin myofilaments, and your myosin, which is your thick myofilaments. Your contraction inside of the myofibril, which are the organelles inside, actually occurs from Z to Z. So these are contracting, these are contracting. But when we're talking about this, we're talking about one sarcomere. Okay, so just to kind of see what it looks like when it's once you're inside. So once you're inside of your, so action potential is occurring here. This is your neuromuscular junction. So action potential, due to the fact that ACH has crossed the synaptic cleft and attached to your receptors, and sodium and potassium ion movement has occurred, action potential is going to propagate, which means move into your muscle fiber. That is going to go, this is called your T-tubule. It's just a tunnel. So action potential will actually go through this tunnel, and it's going to cross this organelle called the sarcoplasmic reticulum. If you guys remember rough ER and smooth ER, endoplasmic reticulum, that's what that is. But in the muscle, it's called sarcoplasmic reticulum. They contain a different ion called calcium. They are just like this ion, or this organelle called the sarcoplasmic reticulum has all these calcium ions. And when action potential comes through, it's going to release the calcium. And the calcium will go below and actually attach to your sarcomere. And that's what's going to cause the muscle contraction to occur. But I'll get more of that in a minute. Each myosin consists of two twisted protein strands called cross bridges that project outward. The myosin head will contain ATP. ATP is the energy that you need. So not only do you need the calcium that comes in and attaches, that comes from the sarcoplasmic reticulum, you also will need ATP that's constantly being produced by the mitochondria. Actin consists of a double-stranded helix. I'll show you that in a second. Called troponin and tropomyosin. At rest, the troponin and tropomyosin actually are blocking. So if there's no calcium, it's almost like having the parking brake on. Your myosin cannot attach, and therefore muscle contraction cannot occur. So just to talk about the structures, so this is the actin and this is your myosin. So we're just really zoomed into a sarcomere right now. So the Z to Z, just imagine that we're really zoomed in to that sarcomere, and then we have our actin and our myosin. The actin, the thin myofilament, is made up of troponin. Troponin is this globular structure here. Tropomyosin, which is the string like double helix, okay? And then you've got the myosin. So your myosin is this thick myofilament, and it has a myosin head. So what's going to occur is when action potential comes in and releases the calcium will actually attach to this troponin structure, and when that happens, it pulls the parking brake, and the tropomyosin will actually move and expose the binding sites. And then as long as this has ATP to get the energy, it will come up and attach and move. And then as much as calcium comes in, it'll keep moving, keep moving. What causes the calcium to come? Action potential. What causes the action potential? Your thought. So as long as you want to move, it will continue to occur. Once you're done with that conscious control, no more calcium will come and your muscles will not contract. So that's what this says. The sarcoplasmic reticulum is in the endoplasmic reticulum of the muscle and contains a high amount of calcium. This is the third ion we've talked about, so just make sure you have them straight. Once the contraction occurs, the calcium will bind to the troponin, changing the shape and altering position of the tropomyosin. The movement of the tropomyosin molecule exposes the binding sites of the actin, allowing links to occur, and that's when you have a cross bridge. The sarcomere throughout the muscle will therefore shorten, and you now have contraction. Just to show you what the sarcoplasmic reticulum looks like, so just imagine action potential occurring, going down the T-tubule, and when that happens, it almost like covers all of your, so like every myofibril is covered by the sarcoplasmic reticulum, which is covered in calcium. So as soon as action potential goes through the little tunnels, it's going to release all the calcium, and it's going to go in and attach to the troponin, unwinding the tropomyosin, and they just start to contract. So again, this is the other diagram I showed you, but it goes down the T. So action potential goes down the T-tubule, releasing the calcium from the sarcoplasmic reticulum. The calcium comes in and will attach to the troponin, unwinding it and exposing the binding sites. Okay, so just so you know, the sliding filament theory this is very important, number 10. I'd put a star next to it or highlight it because it's a definition of what the sliding filament theory is. It's when your sarcomere shortens, the thick and thin filaments slide past each other. So they just slide past one another. The H zone and the I band will shorten. So this will be like a relaxed, and this would be a contracting. You can tell because your H zone is very, very small. So just another diagram. So just imagine action potential, right? ACH is attached, sodium's coming in, potassium's coming out, and all that energy, the action potential, is going to propagate through the T-tubule. And when it does that, it's going to cross that sarcoplasmic reticulum, releasing the calcium, and it's going to come down and attach to the troponin, unwinding the tropomyosin, which is the string one, exposing the binding sites, and now we're going to get into ATP. So at the same time, so you have to have ATP because you need energy. The myosin head is doing a lot of doing the work. So ATP is going to break on the myosin head, and it's going to come up and attach to that binding site and pull it towards the middle or the M line. So you have to have that ATP to be broken down to ADP plus P, or action or contraction cannot occur. So two things you really need. You have to have your calcium and you have to have your ATP. So not only do you have calcium coming in, attaching to the troponin, but you also have ATP attaching here and breaking to form ADP plus P. When you break a bond, you get energy, and the myosin head will absorb that energy and come up and move it. So calcium from the sarcoplasmic reticulum causes the head of the myosin to split ATP to form ADP plus P, giving the myosin head energy. The myosin head will attach, forming a cross bridge. The myosin head rotation will rotate the sarcomere towards the center. That's called the power stroke. The myosin head will then detach, rebinding ADP plus P to form ATP. As long as you have calcium and ATP, the head of the myosin will reattach another binding site further down and keep pulling. So you're getting closer and closer and shortening that H zone. And that essentially is muscle contraction. So just showing you again, just imagine ATP here breaking to form ADP plus P. Remember, in order for that to happen, calcium had to have come here, attached, unwound tropomyosin, exposing the binding sites. So another picture to show you. So just imagine calcium right here. So this is step one. Okay, so step one's from here to here. Imagine calcium attaching here, and use your ATP on the myosin head. So once calcium's here, it's going to unwind, expose the binding sites. ATP will break to form ADP plus P. When you break the bond, it will absorb the energy, come up and create a cross bridge. It will then do a power stroke, meaning it moves the actin towards the M line, or slides it past the myosin. And then it will reattach ADP plus P, and as long as more calcium comes, it will keep happening. Okay, so that's it for now. We'll talk a little more about resting potential next.